Hey guys, welcome to the second part of the Kaplan enzyme series video, biochemistry. I don't know what to call it, but I did a couple of changes today. I have gotten a Blue Yeti mic because maybe just trying really hard to improve the audio quality will make these better because I want you guys to experience the best experience possible. But yeah, let's try it out. This is like I have a camera, I have a microphone, I have my iPad, and then I have my laptop. Just everything is happening all at once. But regardless, today we're going to be finishing up the enzymes chapter so we can move on to the next chapter, which is going to be non-enzymatic protein. It's going to be kind of a drag. Some of these chapters are kind of a drag, but hang in there. They're all important. Don't worry about it. Don't fret. Just push through, okay? You got this. I have faith in you. All right, so this is 2.2 to 2.5. We got enzymes. So to begin with, we have to talk about the enzyme substrate binding. So what is an enzyme and what is a substrate? So an enzyme is the molecule upon which an enzyme acts. That's pretty much all there is. It's like the thing that goes into an enzyme and the enzyme does this fancy chemical reaction. The enzyme substrate complex is the physical interaction between the enzyme and the substrate. So obviously the enzyme and the substrate have to physically interact for a chemical reaction to occur. Has to be a certain conformation, has to be a certain specificity and orientation of the substrate for the chemical reaction to occur. And what's really, really important on this page is the active site. So the active site is the location within the enzyme where the substrate is held during the chemical reaction. Now that's really, really important to know because when we talk about active site and allosteric sites, those are two different things. The active site is where the chemical reaction takes place, and later we're going to find out that the allosteric site on the enzyme is where modifications can happen to the enzyme. The conformation of the enzyme can be changed by inhibitors or activators that can change the availability of the active site for the enzyme to act on the substrate. So here, just a quick image. Here we have a substrate, we have an enzyme, we have an enzyme substrate complex. So on the next slide, we're probably going to talk about the induced fit model and the lock and key model. It'll all make sense when we talk about it. So it's theory time. Woo! Yay! Theories. Once upon a time we had to come up with a reason or a way that these substrates even bind to our enzymes. So we came up with two reasons. We came up with the lock and key theory, and we came up with the induced fit model. Now, the lock and key theory is pretty much how it sounds. You got a lock and you got a key that fits exactly into the lock with the exact conformation that the lock is. So the lock and key theory suggests that the enzyme's active site, the lock, is already in the appropriate conformation for the substrate, which is the key, to bind. No alteration of the tertiary or quaternary structure is necessary upon bonding of the substrate. So basically the key and the lock have a shape that comes together already. They don't need to really change their shapes to fit together. They're just already meant for each other, It's like happy ever after, love, whatever. Now the induced fit model is something that I really want you to know. I want you to know both, but the induced fit model is extremely, extremely important on this page because, I don't know, you might be asked about it. So the induced fit model says the substrate and the enzyme both change shape to fit, complement each other. This model starts with a substrate and enzyme active site that don't seem to fit together. So you have an enzyme and a substrate that initially don't really fit together that well, but when they're binding to each other, they mesh and they change shape to lock into each other and to really fit together. So this is the model that we want to know, that we believe in. The lock and key theory is a little bit too simple. But moving forward, a way to think about it is our good friend, the stress ball. Here we have a yellow stress ball, and we got a hand holding it. I'm not quite sure how I laid this out on this page. So the enzyme is a stress ball. Cool. And the substrate is the frustrated MCAT student's hand. So that's us, right? We're completely frustrated. So the student squeezes the stress ball to reduce stress and relax. a student's hand squeezes both ball, enzyme, and hand substrate change shape. And again, a change in shape in biochemistry, we say change in conformation, to fit each other after the formed stress ball enzyme returns to its original shape. So this is kind of the induced fit, right? You got your hand, which is a substrate. You're squeezing it. I mean, initially, the ball, you're not really getting your stress out, you got to squeeze that ball to get the stress out. So once you start really squeezing it, you just fit together with that stress ball, you really get your anger out. And you're like, darn, I hate the biochemistry section of the MCAT section. And then once you're done, once the reaction's done, the chemical reaction, you let go slowly. And the stress ball just goes back to its regular shape. It's pretty crazy, I know. If that didn't help, you don't have to think of it that way. It's a pretty easy definition for induced fit. But yeah, we got a good friend, the stress ball. Anyways, moving forward, enzymes have helpers. And by helpers, I mean they have molecules that help them perform chemical reactions. So we're going to talk about cofactors and coenzymes. So a cofactor is an inorganic molecule or metal ion often ingested as dietary minerals. So we take it, as a dietary mineral, take it part of our diet. A coenzyme is a small organic group, the vast majority of which are vitamins or derivatives of vitamins such as NAD+, FAD, and coenzyme A. So cofactors are inorganic and coenzymes are organic. Now, both are non-protein molecules that are small in size and bind to the active site of the enzyme to help in catalysis. And catalysis is basically just what the enzyme's doing, its action, its chemical reaction, right? So ionization, protonation, deprotonation, enzymes do a lot of things. And we'll see that down the line. When we talk about some of the pathways of glycolysis and the citric acid cycle, it's going to be super fun. It's going to be amazing. But something I want you to know, on top of knowing what a cofactor and a coenzyme is what an apoenzyme and a holoenzyme is. This is extremely important. I did not think I had to know this, but it was mentioned on my exam. So I would say know everything. Just have like a good idea, especially when there's like concrete definitions to things, know it. Here we have an apoenzyme, and an apoenzyme is an enzyme without their cofactors. So another way of thinking about an enzyme without its cofactors, it's not a complete enzyme in its active site, like it can't perform its function without its helper, the helper's not there. So you can think of an apoenzyme as an inactive enzyme because it doesn't have everything that it needs. Now, a holoenzyme, on the other end, is an enzyme with their cofactors. So if you got your best friend with you, you're complete, you don't got to worry about nothing. You're active, you're good, you're all set. So holoenzymes are active, apoenzymes are inactive. Now, something else we want to talk about that was briefly mentioned in the chapter is what a prosthetic group is. So prosthetic groups are tightly bound cofactors or coenzymes that are necessary for enzyme function. So here we have a cofactor in the active site. We have the active site, the coenzyme, the holoenzyme, the apoenzyme. And we have a prosthetic group, which we'll see with hemoglobin. We'll see that hemoglobin needs an iron prosthetic group in order to bind oxygen. But it's just something to have in the back of your mind right now. I guess I just put in this diagram to like show you where the cofactor is. I'm not 100% sure. For a second, I thought I put it in there for the prosthetic group, but apparently I didn't. So moving on. So do you remember how we said vitamins are coenzymes? They're these organic molecules. Well, we got to briefly know what the fat-soluble vitamins are and what the water-soluble vitamins are. Now, this seems ridiculously intimidating because, for one, it says step1.com on the bottom, and two, it's just like a lot of letters and a lot of numbers and a lot of words. And I don't like that all at once. I know we're taking the MCAT, but I still don't like that all at once. So I got an easy way for you to remember it. So fat-soluble vitamins are A, D, E, and K. A deck, right? Just remember A, D, E, and K. For some reason, those 4 letters just stick in my brain a little bit. I'm not completely sure why. A deck, just remember it. And then everything else is the water-soluble vitamins. Boom, done. That's all you gotta know. Moving on. All right. Now, if what we've already talked about is not already super important, enzyme kinetics is super important. There were so many questions about enzyme kinetics. I want you to know it like the back of your hand. Now, before we start, I'm going to talk about enzymes and how they function. So from now moving forward, I want you to think of enzymes as biological catalysts. They're biological proteins they're biological proteins that perform chemical reactions on a substrate, on another molecule. And they have a velocity. There's a reaction velocity of enzymes. So, for example, let's go back to the stress ball example, where we have a stressed MCAT student's hand. And we got the stress ball, and we got the hand. So here we have the hand and the stress ball. And we have 2 hands right now. Right? So the enzymes, these enzymes right here, can only de-stress 2 MCAT students' hands per second. You can't really de-stress anymore because you only have 2 hands. So in this case, if we say the ball is the enzyme, we have a lot more balls, we have a lot more enzymes than we do hands, and the hand being the substrate. So in this case, enzyme concentration is much higher than substrate concentration. Now, if we move down, we now realize that we got a lot of stressed out MCAT takers, right? There's a lot more hands. We got 6 enzymes, but we got 5 hands. We got 5 substrates. Now, our reaction velocity for this is going to be 5 hands per second because we got 5 hands being de-stressed per second. And now we've reached saturation, right? Every single enzyme that we have in our reaction has been occupied. The active site has been occupied. We got 6 hands per second. That's a lot of hands. A lot of MCAT students de-stressing from this exam. But what happens? So we said that we reached maximal velocity up here, right? Maximum reaction velocity for our enzymatic reactions. What happens if we add more hands? What happens if we add more substrates here? Will that make a difference? Will that increase our velocity? No, it wouldn't, because we have occupied all of our enzyme active sites and have reached our maximal velocity, our Vmax. All of our enzymes are saturated. They're all taken up. We don't have any more. The only way we can increase our maximal velocity at this point is if we add more stress balls, if we add more enzymes to the reaction. So what I want you to think of is, in this case, we have saturated our reaction with substrate. Saturated, when you hear saturated a reaction, that means we've added enough substrate to reach maximal velocity for the enzymatic reactions that are occurring. So here, just to get the point across again, adding more substrate at Vmax will not increase the reaction velocity. At Vmax, you must add more enzyme to increase reaction velocity. So here we have a reaction velocity, right? So we have like an enzymatic reaction, and we have substrate concentration. We see that as we add substrate, our reaction velocity is increasing, but then we reach a point where all of our enzymes are just taken up. You know, they're all used up. We don't have any more space to react with more substrate. So we've reached this Vmax point. And this is called Michaelis-Menten enzyme kinetics. I just want you to know as enzyme kinetics for now. It's not too big of a deal. So a good way of thinking about how enzymes interact with substrate is we have an enzyme and a substrate first, enzyme plus substrate. And then we have a rate of formation of the enzyme substrate complex, which is just basically where the substrate's bound to the enzyme. It's inside the active site. We also have a K-1 rate, which is once the enzyme substrate complex forms, it can go backwards and release the substrate, so it's just the enzyme and the substrate separately. And then we have a KCAT, which is the enzyme plus the product. Now the enzyme has reacted on the substrate and it's formed the product. And under these conditions, the enzyme concentration is kept constant. Under enzyme concentration constant conditions, we can follow the Michaelis-Menten equation. Now, this is the Michaelis-Menten equation. I'm not going to lie, when I went into the exam, I didn't have it memorized. But it's good to have it memorized. I just didn't. I would have it memorized. It's a good thing to know, but what I would more focus on instead of memorizing the Michaelis-Menten, it could be asked as like, what's the Michaelis-Menten equation? Sure. But what's better is you understand the variables. You understand what Vmax is, you understand what Km is, and you understand what this is, like substrate concentration. Once you understand what it is, it makes more sense as to why this equation is this equation. So something I want you to know is very, very important. What is Km? We've talked about what Vmax is. We've talked about what the substrate concentration is. What the heck is Km? So the Km is when the substrate concentration is the Km is the substrate concentration when the reaction rate is 1/2 maximal velocity. So let me go back up here. So here we said this is Vmax, right? This is when all the enzymes are taken up. The Km is right here. So the Km is the substrate concentration, right? Right here. We've added this much substrate. And when we've added this much substrate, our reaction velocity is 1/2 Vmax. So Km is going to be a substrate concentration. Km is not a reaction velocity. It is a substrate concentration. Very important to know. What I would know is Km equals substrate concentration when reaction rate is 1/2 Vmax. Very, 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 very important to know. And that's called the Michaelis constant. So again, Km is the substrate concentration at which half the enzyme's active sites are full, half of the stress balls are in use. So if you think about the stress balls, we got half of them in use. I'm not the biggest fan of the stress ball analogy, by the way. I'm just continuously saying it because, I don't know, I have it part of this, I guess. So we said Km is a substrate concentration at which half of the stress balls are in use or half of the maximal velocity has been reached. So if we think about an enzyme, right? If we have an enzymatic reaction and those enzymes need like a little bit of substrate concentration, you add a teeny little bit of substrate and you've already reached half maximal velocity. Well, you could think of that as the enzymes and the substrate have high affinity for each other. They really like binding to each other. It's really easy for them to come together and to start working together. But if you need to add like a ton and a ton of substrate for an enzyme to start really ramping up and to really start like working hard, then, well, they're not really that great together because you have to add so much substrate for the enzymes to have half maximal velocity, for the active sites to be taken up. So Km is actually a measure of affinity between the enzyme and the substrate of that enzyme. So here, measure of an enzyme's affinity for its substrate. Km is an intrinsic property, so it does not change based off of any variables. It is intrinsic of the enzyme. It cannot be affected by changing enzyme concentration or substrate concentration. So no amount of enzyme that you add, no amount of substrate that you add is going to change the Km of the enzyme. Pretty cool. So again, it's so important that I mentioned it again. Km equals the substrate concentration needed for 50% enzyme saturation. Again, remember when I said saturation means all the enzymes are taken up? That's where we mention it again. So here we have low Km equals the enzyme has high affinity for substrate, right? Because you have to add like a teeny bit of substrate to reach half maximal velocity of the enzyme. The enzyme loves the substrate, right? It just grabs onto it as soon as they see it. If you could add so much substrate for the enzyme to do its thing, it's got low affinity. Enzyme has low affinity for substrate. So when the substrate concentration is less than Km, changes in substrate concentration will greatly affect the reaction rate. So this is like a Michaelis-Menten curve for an enzyme, we see that early on, when we have like a little bit of substrate, the more substrate we add, it has like an exponential like effect on the reaction velocity of the enzyme. But when the substrate concentration is greater than Km, right? When we're above this Km, the reaction rate increases much more slowly as it approaches Vmax, where it becomes independent of substrate concentration. So this is the Michaelis-Menten relationship graph. It's a hyperbola, right? Some of our enzyme kinetics that we're going to talk about that aren't Michaelis-Menten, they're cooperative enzymes like hemoglobin. They're going to be sigmoidal. They're going to look like an S. So we're going to be going through some different enzyme kinetics and graphs. But what I want you to know for now, for this lecture, is that when the substrate concentration is below Km, the reaction velocity increases really, really fast. But when the substrate concentration goes past Km, the reaction velocity increases much more slowly until it reaches Vmax, flat lines, right? Where no amount of substrate that you could add is going to change the Vmax. But what will change the Vmax? That's right, you got it. Add more enzymes. Boom. Good stuff, guys. I knew you could do this. So, some more variables to keep in mind. Vmax is the maximum enzyme velocity, moles of enzyme per second. Now, I just want you to know that the Vmax is the maximum enzyme velocity. Moles of enzyme per second, not too important. Vmax equals the enzyme concentration times kcat. I would remember that because it may be important, but it's not necessarily something that is super duper important. I think Km is much more important, but I would commit this to memory. Kcat is the number of substrate molecules turned over or converted to product per enzyme molecule per second. So it's like the catalytic speed. How many substrate molecules are turned into product per enzyme molecule per second? There's not really much else you need to know about that. But this is a relationship that I would have. And kcat has units of 1/seconds or the inverse of seconds. But what I want you to really, really know is this relationship. I have it highlighted. I love it. This relationship will come back over and over again, not only in your biochemistry class, but also during the MCAT. The catalytic efficiency, which is kcat over Km, basically tells you how good an enzyme is, how amazing it is at what it does. So we got the turnover rate, right? We said kcat is how many substrates the enzyme turns over to product per enzyme per second. And then we have Km, which is the affinity. So the substrate affinity for the enzyme. How much does the enzyme love the substrate? This ratio, the catalytic efficiency, tells us the efficiency or how good the enzyme is. And it is highly tested in the MCAT. The higher the Km, or the higher the catalytic efficiency, the better the enzyme. Now, how can we remember this? What if I don't know, right? What if I know what catalytic efficiency is, but I don't remember that's kcat over Km? How do we commit this to memory? Well, let's think about it. An enzyme is really good at its job if it can really have a high affinity for its substrate, right? If it can really love its substrate and bind to it, even if there's little amounts of substrate, it's a pretty good enzyme. So think about it. If our Km is lower, it's in the denominator, which means that the fraction is going to be larger. If our kcat, which is how quickly our enzyme can work, is higher, that means it's a better enzyme. So kcat is higher, Km is lower, which means that our catalytic efficiency is only going to get better as the enzyme gets better. Bada bing, bada boom. You know, all you got to know is the kcat and the Km. You have to know the two variables involved, and you can reason through the proportionality, the relationship between the variables. So now we get to talk about our lovely Lineweaver-Burk plots. Lineweaver-Burk plots are the inverse of these graphs. So we talked that this is the Michaelis-Menten relationship right here. We got reaction velocity. We got substrate concentration. What Lineweaver-Burk plots is the inverse of Vmax and the inverse of Km on a graph. And the reason we love Lineweaver-Burk plots over these sorts of graphs is because right here we can't really tell where Vmax is. How do we tell what number Vmax is on? We don't have a concrete quantitative number, because this line, this hyperbola, just goes up and it kind of flatlines. It kind of turns into a limit, or turns into a unknown when it hits that Vmax position. So we want to have concrete numbers for the Km and the Vmax. So if we take the inverse of both, we start getting a Lineweaver-Burk plot. So it's a double reciprocal graph of the Michaelis-Menten equation. We use it because we got a straight line, and we can work with the straight line. We can find what the Vmax is with a straight line, and we can find what the Km is with a straight line, and our slope also tells us something. So here, this graph, I don't know why I scrolled all the way back up, because I have it right next to the Lineweaver-Burk plot. But here we have the Lineweaver-Burk plot. We have a straight line, and what I want you to know is the x-intercept of this line is negative 1 over Km. I want you to know that the y-intercept of this line is 1 over Vmax. And I want you to know that the slope of this line stands for Km over Vmax. Those are 3 important things to know, because later on we're going to talk about enzyme inhibitors. So molecules inhibit enzymes, and they change the Km and the Vmax of the reaction. So we're going to be able to tell if we have an inhibitor, what type of inhibitor do we have for our enzyme. So remember I talked about cooperativity, I talked about hemoglobin, and I said that hemoglobin has this sigmoidal shape instead of this hyperbolic shape that the Michaelis-Menten graphs have. Well, certain enzymes don't show the normal hyperbola when graphed on a Michaelis-Menten plot, which is going to be reaction velocity versus substrate concentration, but rather show sigmoidal S-shaped kinetics owing to cooperativity among substrate binding sites. So cooperative enzymes are enzymes that when the substrate binds to one active site, so they have multiple active sites, they have multiple subunits, when the enzyme or the substrate binds for example, let's take hemoglobin as an example. When oxygen binds to one of the active sites of hemoglobin, right, let's say we have 4 active sites, it increases the likelihood of oxygen binding to the other active sites, because the affinity between those active sites and oxygen increases as oxygen binds to one active site, to 2 active sites, affinity gets more and more and more for the enzyme for the oxygen. So obviously we're going to have a different shaped Michaelis-Menten curve. We're going to have a sigmoidal shape. We're going to have cooperativity. So sorry about that. My camera just quit working for a little bit. So back to what we're talking about. Cooperativity can be thought of as an enzyme having multiple active sites. And when the substrate binds to one of the active sites, the other active sites cooperate and increase their want or their affinity for the substrate. So from that logic, cooperative enzymes must have multiple subunits, because you need multiple subunits. You need multiple protein quaternary structures in order to have multiple active sites for the enzyme. And you need more than one active site. Let's see what we talked about. So these cooperative enzymes have a low affinity state and a high affinity, relaxed state. So we call the low affinity state tense, and we call the high affinity state of the cooperative enzyme relaxed, or R. So the tense state is as one subunit loses substrate, other subunits are more likely to also lose their substrates. They lessen their affinity for the substrate. High affinity relaxed state, as one subunit binds substrate, other subunits are more likely to also bind the substrate. So we have a tense state, this is hemoglobin, for example. We have a tense state. We don't have any oxygen bound to any of our we have 4 subunits here of hemoglobin. We don't have oxygen bound to any of them. And then we have an oxygen bound to one, increases the affinity for these 2. Then we have oxygen bound to second, increases affinity. Oxygen bound to third, increases affinity. Oxygen bound to all four, the relaxed state. So you see how the red turns into purple? That means they're more likely to bind oxygen. They want oxygen more. And that's that cooperativity. Something that I want to mention that could be mentioned in some of your passages on the MCAT is what the Hill's coefficient is. When I went into my exam, I didn't know it, so I want you to know it. The Hill's coefficient quantifies cooperativity of an enzyme. Basically it defines like, is an enzyme cooperative or is it not? Or is it negatively cooperative? Where if one substrate binds, it actually decreases the affinity of all the other substrates for the substrate. So if you have a Hill's coefficient greater than 1, it's a number, you have positive cooperativity. If you have a Hill's coefficient less than 1, you have negative cooperativity. And if you have a Hill's coefficient equal to 1, we have no cooperativity. So that enzyme would not show cooperativity. So now we're going to talk about the effects of local conditions on enzyme activity. This should be pretty basic, should be pretty quick. Basically, the activity of an enzyme is heavily influenced by its environment. So what environmental factors would an enzyme, a protein, have to deal with in the body? The temperature, the pH, and the salinity, the saltiness. So the enzyme activity equals the enzyme velocity equals the enzyme rate. I want you to know that all three of these are synonymous on the MCAT. So you can see enzyme velocity, enzyme rate, enzyme activity. They're all the same thing. So enzyme catalyzed reactions double in velocity for every 10 degrees Celsius increase in temperature until the optimum temperature is reached, after which activity falls off sharply. So the enzyme can denature at higher temperatures. So enzymes are these catalytic machines that have an optimum temperature at which they function, an optimum temperature at which they do their job the best. So for enzymes in the body, the optimal temperature has to be 37 degrees Celsius because that's the normal temperature for the human body. So obviously enzymes that function in our body have to have an optimal temperature of this. Now, for example, enzymes that are in our stomach, they have to function at lower pHs. They have to function in really acidic situations that enzymes in other parts of our body don't have to. So that's how it's really important to know. So pH affects the ionization of the active site. Changes in pH can lead to denaturation of an enzyme. That's why pH is important. Optimal pH for enzymes that circulate the human blood is 7.4. And 7 is chemically neutral. So there's a difference between physiological neutral, which is in our body, and there's a difference between chemically neutral, which is, when we talk about chemistry, we have 7 as the neutral pH. But in our body, 7.4 is physiologically neutral. So when you think of chemistry, think of 7 as like chemically neutral. But when you think of biochemistry, when you think of biology, when you think of the human body, think of 7.4 as the pH that is physiologically neutral. So exceptions to pH 7.4 in the body, we have pepsin enzyme in the stomach that has a maximum activity as pH 2. So remember, our stomach is really acidic. The enzymes like pepsin that function in our stomach are going to be functioning optimally at lower pHs, at more acidic pHs. We also have pancreatic enzymes that work in small intestine, that work at actually more basic conditions, so at pH of 8.5. Here we have relative enzyme activity and pH, relative enzyme activity and temperature. You see how as the pH increases, the relative enzyme activity for these two enzymes, this one functions the best at what looks like about 2.5 pH maybe. This one functions the best at the maximum activity at what seems like about 7-ish pH. Temperature can also have the same effect. This enzyme and this enzyme are different. This one functions the best. It has its peak at about like 15 degrees Celsius. This one had its peak at about 37 degrees Celsius or something. A couple of other definitions that I want you to know. What in vitro and in vivo means. In vitro means in a test tube, and in vivo means in a living organism or the body. And we mentioned how salinity also has an effect on enzyme activity. So salinity and osmolarity is not generally of physiological significance, but altering the salt concentration can change enzyme activity in vitro. So in vitro being in a test tube, generally we don't really consider salt when it comes to the body because we have kind of like a baseline level, and we have a way of filtering it. But in test tubes, we've seen that if we add way too much salt or way too little, whatever, it disrupts hydrogen bonds, ionic bonds. So those are the two bonds. All right, cool. That was all that there was about that. And now we can talk about the regulation of enzyme activity. This is also extremely, extremely important. But my camera is running out of charge, so I suppose I will save this for another video. Okay, so this is 2.5 to what looks like, this is the final portion of the video. I guess I'll just do it. Actually, let's just do it. Let's try to get through it. So we have ways of turning enzymes off. We don't want enzymes to constantly function. We want a way for them to do their job and then to stop. Because if they're overactive, they can really hurt your body. So we have feedback regulation, which are enzymes are subject to regulation by products further down a given metabolic pathway. So there's two types of feedback regulation. We have feed-forward regulation, where enzymes may be regulated by intermediates that precede the enzyme, that come before the enzyme in the pathway. This is much less common. What's very much common in our bodies is the negative feedback. So negative feedback is feedback inhibition, where once we have enough of a given product, we want to turn off the pathway that creates that product rather than creating more. So basically, our enzyme uses substrate, creates product, and that product comes back that it just created and turns the enzyme off. It's like, hey, we got enough of it. We don't need any more. This is a good example. This is hormonal. In biology, we'll talk about hormones. It starts getting a little bit complicated, but long story short, the hypothalamus produces this releasing hormone. This releasing hormone goes to our pituitary gland and releases another hormone that acts on our organs, our adrenal cortex, and that forms glucocorticoids. Now, these glucocorticoids negatively feeding back, they go back up and they negatively feed back on not only the pituitary gland, but also the hypothalamus. They're like, hey, we got enough glucocorticoids, we don't need any more. So that's an example of negative feedback. And now we can talk about reversible inhibition of enzymes, and I don't want to rush through this, so if I have to make another video for it, I will, because these are extremely, extremely important. We have competitive inhibition, we have non-competitive inhibition, mixed inhibition, and uncompetitive inhibition. And you have to know all of them, because really, even if the book says like you only have to know competitive and non-competitive as the main ones, I would know all of them like the back of your hand, know them going into test day. So for competitive inhibition, we have these molecules that bind to enzymes, this inhibitor that binds to an enzyme. And for competitive inhibition, the inhibitor binds to the enzyme's active site to stop it, to inhibit it. And what we have to know is, how is Vmax and Km changed? For competitive inhibition, because our inhibitor binds to the active site, if we add a lot of substrate to the mix, we can overcome that effect. Because if we add a lot of substrate, if we overwhelm the reaction with it, then there's more likelihood that the substrate will bind to the enzyme active site instead of the inhibitor. So again, Vmax for this would not change, because if we add enough substrate, our enzymes can function at maximal velocity, at their highest peak. But our Km is increased, because if you remember, Km is a substrate concentration which we have half maximal velocity of enzyme. We need more substrate to reach half maximum velocity, because this inhibitor is like making it hard for us to reach our peak potential for our enzymes. So here you have to add more substrate to overcome the inhibitor. So what you have to remember is Vmax, no change, Km increased. Now we have non-competitive inhibition, which is our inhibitor, which is this red dot here that I've made, this random molecule, it binds equally to our enzyme and our enzyme substrate complex. So it binds to both with the same affinity, but it does not bind to the active site of the enzyme or the enzyme substrate complex. It binds to an allosteric site. It binds to a site that is not the active site, a site besides it that modulates, that modifies the active site when it, or the shape of the protein, and modifies the accessibility of the active site. So for non-competitive inhibition, the Vmax is decreased because we have less enzyme to work with. Once our inhibitor binds to our regular enzymes, it basically defeats the functionality of that enzyme. You can't use it anymore. So if you decrease the amount of enzymes you have, you're going to decrease the Vmax. You're going to decrease that maximal velocity. But Km doesn't change because the enzymes that we already have, that are already functioning, that are already working, that don't have any inhibitor bound to them, they can function just normally fine. They're completely fine. So our Km would not change in this case. Now, mixed inhibition is different. We have a mixed inhibitor that binds to the allosteric site of the enzyme and the enzyme substrate complex, just like in non-competitive inhibition. But the only difference is the inhibitor binds with different affinity for the enzyme or the enzyme substrate complex at the allosteric site. If the inhibitor bound with the same affinity for both, with the same want, it would be a non-competitive inhibitor. But in this case, it's a mixed inhibitor. So the inhibitor binds to enzyme allosteric site, but with different affinity for the enzyme versus the enzyme substrate complex. So for a mixed inhibitor that has a greater affinity for the enzyme, it's going to increase Km and it's going to decrease Vmax. What I want you to know from mixed inhibitors is Vmax is always decreased. Km, for a greater affinity for the enzyme, is going to increase Km, right? Because we're going to need a lot more substrate for the reaction to reach half maximal. We're like eliminating enzymes from the equation. What I want you to think of when a mixed inhibitor has a greater affinity for the enzyme substrate complex is that if you lock in the enzyme and the substrate together, you're basically increasing their affinity. They like love each other more. So you're going to be decreasing the Km in this situation because you're increasing affinity, and you're also going to be decreasing Vmax. In both scenarios, you're decreasing Vmax, but if the inhibitor has a greater affinity for the enzyme, you increase Km. If it has a greater affinity for the enzyme substrate complex, you decrease affinity, the Km. So then we have uncompetitive inhibition at the end. Basically, inhibitor binds to an allosteric site of only the ES complex, the enzyme substrate complex. So once the enzyme and the substrate come together, they bind to each other, it opens up a position on the enzyme that the uncompetitive inhibitor can bind to. And that's the only time when the uncompetitive inhibitor can bind. But you have to know about this as it decreases both Km and Vmax. So here are the Lineweaver-Burke plots for them. Again, I don't know, my camera's going to go out of charge, so I can't go into this too much. I will make another video talking about this, about the Lineweaver-Burke plots. But for now, I want you to remember that the y-intercept is 1 over Vmax and the x-intercept is negative 1 over Km. So what we just talked about, all the different inhibitors, we have an uninhibited enzyme, and we can tell what type of inhibitor we have based off of what our line looks like on our Lineweaver-Burke plot. So this is important because during the MCAT, you're going to have, they might have one of these graphs, and you have to tell, is it a competitive inhibitor, uncompetitive, non-competitive, that's added to the mix. Finally, irreversible inhibition. Don't have to know it too much, but if you see irreversible inhibition, all the other ones that we talked about were reversible, which means that the inhibitor can pop off of the enzyme, it can leave. Irreversible is basically when the enzyme is permanently altered due to the inhibitor, and you need to make new ones, right? You can't use that same enzyme anymore. You have to make new enzymes through transcription and translation. I'm going to skip over the allosteric sites. We talked about an allosteric site is the site on an enzyme that is not the active site, that regulates the active site's availability. Allosteric enzymes are enzymes with multiple binding sites. allosteric activator is it binds to that site, the allosteric site, and it changes the conformation of the active site. So it makes it more available for substrate, makes it easier to bind. Allosteric inhibitor does the opposite. There's covalent modification of enzymes. We can phosphorylate enzymes. We can dephosphorylate enzymes. We can glycosylate enzymes. Glycosylation is basically adding a carbohydrate molecule to the protein, to the enzyme. Phosphorylation is adding a phosphate molecule, and dephosphorylation is taking off a phosphate molecule. Now, how phosphorylation and dephosphorylation affect the activity of an enzyme is determined experimentally. It's not, you can't determine it by just like knowing a rule of thumb or something. You have to determine it experimentally. Sometimes phosphorylation means activate. Sometimes phosphorylation means deactivate. So, and lastly, after this long video, I want you to know what zymogens are. So zymogens are inactive forms of enzymes. So in our body, we have digestive enzymes. A lot of times in our pancreas, that they need to be in an inactive form because they're released from our body into our stomach or like say our small intestine. And they need to be inactive because if they're not, they're gonna destroy our body itself. They're gonna eat the organ itself, the pancreas, which is not good. We want it to be active when we need it, right? So zymogens are inactive forms of enzymes. And I want you to think of digestive enzymes released from the pancreas that need to be inactive. And then once they're released, then you can activate them once you need them. So zymogens contain a catalytic, an active domain, and a regulatory domain. And the regulatory domain needs to be altered or removed to expose the active site. So that's basically how you regulate these zymogens. And you can tell it's a zymogen because the protein will end in ogen, like trypsin, trypsinogen, pepsinogen. So it's going to be a trypsin, like that'll be the protein, but it's going to be ogen, which means that it's the zymogen, it's the inactive version. And then once you activate it, it's going to be trypsin. So yeah, that's going to wrap up this video. I know it was a really long video. I have to edit this down somehow because my camera gave up on me in the middle of it. But I hope you enjoyed it. And I'll definitely make another video about the Michaelis-Menten, or sorry, not the Michaelis-Menten, the Lineweaver-Burke plots, because I believe this is extremely, extremely important. And also Lineweaver-Burke plots and analyzing them ties in knowing what inhibitor is what and how it functions. So we will definitely do that. But thank you for watching. I don't want to make it any longer than it has to be. And I'll see you in the next video.